Hello everyone and welcome to my project work in CSE 564 visualizations. Uh, this video is part 2 of the project work where we are exploring different visualization techniques like PCA, MDS. We'll also explore some other techniques like scatter matrix plots, Scree plots, and we'll try to understand their significance. Um, just to give a small gist about the project, we'll be working with the red wine dataset from kaggle.com. Uh, this dataset has a lot of numeric fields that explain physiochemical characteristics of the wine. For example, the acidity of the wine, the content of chlorides, the pH values, the amount of alcohol, and finally we have our target variable, which will be the quality of the wine, and we'll measure the quality on the scale of 0 to 10. So starting with the project, let's first explore the project structure. So here is the project structure for part 2. So the project is divided into two major phases, the front end part and the back end part. So the front end part consists of the JS files that are built on D3JS and other visualization libraries, and the backend part is built using Python, which uses all the data science libraries like sklearn, matplotlib for visualization, pandas, and numpy for data manipulation. Um, Now we have our project running, and as you can see, we are generating detailed logs about all the activity in the program, starting from the initialization to the server logs. Um, let's move back to the browser and start with the k-means elbow plot. So k-means elbow plot is a graph that will tell you what is the right number of clusters to choose for clustering the data. So as we can see, it kind of forms an elbow, and at uh, cluster number 4 or 4 clusters, we are getting minimum within cluster error. So we'll go ahead with 4 clusters for stratifying our data set. Uh, Next, we look at Scree plots. Scree plots are a way to measure the intrinsic dimensionality of the data. So uh, each eigenvalue will explain some percentage of the variance in the data, and as we see, about 5 components together are able to explain about 80% of the variance in the data. Um, Next, we look at random data set. Uh, we see a similar graph for random data set where we see about 5 components again explaining about 80% of the data. And finally, we look at the stratified data set that is generated using the k-means clustering and then stratifying into different stratas. Once again, we see a small variation as compared to the original data set, but mostly the trend is almost the same. Okay, so once we are done with the Scree plots, next we'll explore principal component analysis. Uh, so PCA is a technique that performs linear transformation of data points to another coordinate system, uh, at the same time trying to keep the maximum variance of the data intact. So let's look at the PCA for original dataset. So as we know, the dataset had about 11 attributes, and visualizing 11 dimensional data is not possible. So we do a dimensionality reduction using PCA, and we try to project that data into 2 dimensions. So here you see top 2 PCA components that are explaining the maximum variance in the dataset. The points here show clusters of different qualities. For example, wine points with 7 and 8 are shown here, and points with 3, 4, and 5 are forming a small cluster here. Let's see if we have the same effect from the random data set. So random data set has about 50% of the points as compared to original data set, and once again we see a similar trend where we see two main clusters being formed, points with quality 3 and 4 and points with quality 7 and 8. Finally, let's look at stratified data set. This one is not as distinguishable as the other two, but still we see a small pattern forming for blue points and the red points. Okay, so next we discuss MDS, multi-dimensional scaling. So multi-dimensional scaling is a technique for visualizing data, visualizing high-dimensional data um, on 2 or 3 dimensions. So the way MDS works is by uh, exploiting the distances or similarity between the data points. So we form a similarity matrix between all the points in the data set, and then we try to plot them in such a way that similar points are clustered together and dissimilar points are as far away as possible. So starting with the original data set, and once again we'll try to have different distance matrices. We'll try to explore with Euclidean distance as well as with correlation distance. So starting with Euclidean distance, here you see blue points being clustered at one end and red points being clustered at another end. Looking at correlation distance, once again you see a cluster of blue points together and red points together. The correlation distance will calculate the correlation values between each point, and Euclidean distance will calculate the Euclidean distance between each point in the data set. Next, we try to perform similar analysis on random data set. We have Euclidean distance once again. This time data points are 50% less as compared to the original data set, so we kind of see a clear cluster for blue points and red points. Performing correlation distance on the random data set, we see a good cluster of blue points again and red cluster well separated. And finally we perform the same analysis on the stratified data set, in which case we see kind of good distinction in red points, purple points and blue points this time. Finally, looking at the correlation distance, we see that the red points are clustered on the upper periphery and the blue points are clustered on the lower periphery. Finally, we explore the scatter matrix plots for the data set. So while performing PCA, we need to see what attributes are actually giving us the highest PCA loadings, and based on that we try to find the correlation between those attributes in the dataset. So right now we see that the top 3 PCA loadings were free sulfur dioxide, total sulfur dioxide, and the alcohol content in the wine. And these two are kind of related in some sense, that this one will have additional components being added to it, and we kind of see a good correlation between the first 2 PCA loadings, a linear increase or linear correlation. We also see somewhat small correlation in 2 and 3, and almost no correlation for 1 and 3. So let's explore for the random data set, and once again we see a similar trend for 1 and 2 and 2 and 3. This is with 50% less points, so we kind of see a sparse formation here. And finally exploring for the stratified data set, once again a very similar plot for the data. So that is all for this video. Have a great day, and thank you.